This is a schema of the course of the polypeptide chain, alpha helices and so forth. There's no beta sheets, it's just alpha helices. This is one helix, starts over here. This is the beta chain, starts over here, runs through, runs around, and so forth. This beta chain looks exactly the same, except it's just rotated 180 degrees like this. And the alpha chains are underneath. If one is like this, the other is rotated like that, also 180 degrees. This glutamic 6 is where the abnormality is. Glutamic acid is the normal residue here. Glutamic acid is an acid, so it's polar. It's replaced by valine, which is a hydrophobic group. So there's a hydrophobic spot on this molecule where there is not in the normal molecule. And that hydrophobic spot, hydrophobic meaning nonpolar, oil-like, is the thing that sets off the polymerization reaction. This spot fits into a pocket on a neighboring molecule. So one molecule fits into another like this, and then the next one the same way, and that's why you get the long chain. This shows the way the chain on the left side. Each of these is a chain. This is a double-stranded. We can continue to go up this way. And the diagram that I showed you before, the polypeptide chain folding, simply extends this way, and you have here, as I said, a double strand. This is a picture of, the way the detailed molecular structure of proteins was not known 40 or 50, maybe 50 or 60 years ago. The first direct information came from X-ray crystallography. Two studies, Max Perutz in England discovered the structure of hemoglobin, that is that structure, and John Kendrew discovered the similar structure of myoglobin, which is the muscle protein, which is somewhat like hemoglobin. So this is Max Perutz's original study. The alpha chains are white, the beta chains are black, and you can see the oxygen binding site here and so forth. Obviously now it's known in much greater detail, but this is a historical event of monumental proportions in the history of biology. But the interesting thing is that the beta chains, when you oxygenate and deoxygenate, change their position. They're close together in oxygenated hemoglobin, and they're far apart in deoxygenated hemoglobin. And it turns out that that shift, that isomerization, which means change in structure, that isomerization is responsible for the way oxygen is bound to hemoglobin and makes mammalian life possible. If we had a molecule which didn't do that, you wouldn't have the properties of oxygen binding that you have, and you couldn't get along in this kind of an atmosphere. I'll say a little bit more about that later. In any case, this change in structure associated with the abnormal site is what makes the sickle fibers able to form. So that's basically the structure I want to talk about. The solubility, which is the thermodynamics, the equilibrium part, and it's kind of straightforward. You can put a certain amount of deoxygenated sickle hemoglobin in solution, but when it gets to a certain level, it creates a gel. It's like trying to dissolve too much salt in a glass of water. If you put the whole canister of salt in, eventually some of it's not going to dissolve. That's its solubility level. It's the same thing in principle for sickle hemoglobin. You can only put so much deoxy in before you create a gel. So the kinetics, which is the process of forming the gel, I talked about the homogeneous nucleation. That's the first one in bulk solution. Then further nucleation of new fibers on the surface of existing ones, and then fiber elongation. They form a cross-linked gel, which looks like this. And those cross-links are weak. They're weak interactions. They can be broken. But then other ones can form. It's not like a covalent bond, which takes a lot of energy to break. In addition to these properties, these three reactions which produce the kinetics, there's a delay time. This used to be thought of as the critical issue. Now people are arguing that other issues are critical for pathogenesis, but clearly it's important. And what happens is, in a nucleation process, there's a delay before nucleation occurs. If you've ever heard of people who make rain, if you don't have, you're in an area where you don't have rain, and there is some water vapor up above in the atmosphere, but it hasn't condensed to make rain, they drop ice crystals. They drop sodium iodide or other things as crystals, and the water starts to coalesce in droplets. Those serve as nuclei, and then it starts to rain. It's the same thing here. You have to get a number of molecules together by accident. It's an infrequent accident that molecules get together enough to create the beginning of a fiber, which is called a nucleus. So there's a delay time. And that delay time is very variable depending on conditions that one is operating in. If the delay is longer than the 1 or 2 seconds that the red cells take to pass through the capillaries, which is where oxygen is taken out, then the red cells will go through those small vessels without gelling and will get back into the lungs, and the patient will not be damaged if the delay time is long enough. If the delay time is too short, the vessel will be occluded, will be blocked. So that was considered to be, and probably still is, a very critical issue. What's the delay time? So that's one property. The next property is, if you have something which is a nucleation process and occurs rarely, you can't predict when it will occur and where. If you have a beaker full of stuff, you don't know if it's going to occur over here or over here or over here. It's just an issue of probability. So that means if you repeat an experiment, it won't be repeated in exactly the same way. That's called random, or the scientific word is stochastic. So stochastic reactions are ones that are intrinsically random. And the other two things which are important are the extent of the rapidity and the extent of gelation are highly dependent on the concentration of hemoglobin and highly dependent on temperature. Okay, so if you now do an experiment in which you look at a solution of sickle hemoglobin deoxygenated, actually look at it by turbidity, but it's not important. You can look at it in many different ways. This is a measure of how much gelation there is. And you can see there's a delay time in the beginning, and then it takes off very rapidly, and finally it levels off when you've used up all the hemoglobin that can be turned into polymer. I really said what this said slide, what I said before, but to illustrate it in this slide, all of these 4 things give the same message. Basically, this is what's called the supersaturation ratio. That's the concentration of your hemoglobin divided by the concentration at solubility. So if your concentration is less than supersaturation ratio is less than 1, that is to say the concentration is less than the solubility, everything will be dissolved. If the concentration is more than the solubility, you're going to have something left over which doesn't get dissolved. And the dependence of the delay time on supersaturation is very great. It's a 40th power dependence. That means to say the delay time, or actually the inverse of the delay time, is proportional to the 40th power of the supersaturation ratio. Well, that's a lot. If you have a 1% change in saturation, that's going to produce like a 50 or 60% change in the thing that you're looking at, that is the delay time. So here's the mechanism which puts together the thoughts. I've been trying to present this sort of in a pictorial way, and maybe I should have started off with the picture first and then the details. But this is the picture which summarizes the mechanism. Here's an individual molecule of sickle hemoglobin, deoxysickle hemoglobin, and it gets together with a neighbor, and it hangs together for a little while, but it's unstable, and it comes apart again. And then another one may add on to the two, that is when the two survive, and then you can get another one. That all these things are unstable. The reaction would rather go backwards. But at some point, when you have enough of these you have enough contacts to make it stable. And that's called the homogeneous nucleus. Once that's formed, it doesn't go backward easily. It would prefer to go forward, and additional molecules add on this way and this way to form this final stable fiber. So that's homogeneous nucleation. When you have a fiber, you now can create a second fiber on its surface by heterogeneous nucleation. Hetero simply means other. So this is forming another existing fiber. This is homogeneous nucleation. Homo meaning alone or one. It has to form without any A's. This is simply to illustrate the stochastic nature. This is time. And these are a number of runs on the same solution. Each line represents one experiment, and they're all put on the same scale to see what it's like. So sometimes it starts to gel very quickly. Sometimes it waits a long time before it starts to gel. So you can see the magnitude of the randomness, the magnitude of the stochastic process is very great. that has to be taken into account when one figures out whether it's going to be pathology or not. Okay, if one wants to ask about polymerization, one would also like to know about depolymerization. And this has been a subject which we've investigated and really hasn't been investigated before. How do polymers dissolve? Maybe you could get treatment not just by preventing them from forming, but maybe getting them to dissolve in the most effective way will also be a useful form of therapy, which is what we're interested in. This is fiber length. Lost my pointer here. Oh, here, okay. This is fiber length, and this is time, and this is one fiber. So it starts off at 15 microns in length, and over time, as you measure, it becomes shorter. And these are other ones. So this is a linear process, which is what we would expect it to be, because it loses molecules one by one from its ends. So that means you can measure this rate. Now, if you measure that rate as a function of the concentration of carbon monoxide, which is equivalent to the concentration of oxygen, you find that as you increase the concentration of carbon monoxide, it doesn't make any difference. Something is happening independent of the CO concentration itself. And what's happening is, if you have a fiber here, the ends of it are in dynamic equilibrium with solution. That is, molecules are coming off and going back on. But when they come off, they can't go back on because they get lapped up by the CO and they're not deoxy anymore. So all the CO is doing is serving as a sink, a place to sequester the molecules that came off, and it doesn't make any difference what the concentration of CO is. But now if you go to a higher concentration of CO, you get a different picture. The time for total dissolution, the scale, this is a different thing. This is total dissolution and this is rate. The time for total dissolution, total dissolving of a fiber, is much greater at 0.4 of an atmosphere PCO than it is at a full atmosphere PCO, and it's a relationship that looks like this, more or less hyperbolic. So that says there's another process occurring than simply losing losing molecules from the end. Okay, now I'm just going to digress for a moment. I don't know if you can see this fiber, but this is a fiber which is shortening from its end. There's one end. I'll just leave my pointer there, and you can see that it's shortening. Well, it isn't yet. There we go. Now it's shortening. Now its end is over here, and so forth. That's not very dramatic. But that's the simple way that they can dissolve. But fibers can also do another thing. This is an electron micrograph of a fiber in the process of dilution. And it turns out, in this case, the fiber broke in many places, and each little short fragment shortened. So two processes are concerned here. One is fiber fracture, and the other is shortening of the ends. And you can imagine that will produce much, much more rapid dissolution because you have so many more ends here, and that's where the shortening takes place. So that suggests that maybe sometimes fibers will dissolve completely, and maybe sometimes they won't dissolve completely. This is another verification of that. Here's a fiber which is sort of fading away, and then you can see a break here, enlarged over here. So this is just a verification of what the previous picture showed you, so I won't belabor it. And this is not really relevant, but over here, this is a schema of the time sequence of breaks appearing in a fiber. Each little blip is a break that occurs in the fiber, and over time, you can see you get a lot of breaks, therefore a lot of fragments, and therefore very rapid depolymerization. So schematized, it looks like this. This is dissolution of a fiber from the end. Here are the molecules deoxy. Here are the molecules where they bound carbon monoxide or oxygen. And the fiber just shortens from its end in a straightforward way. But here, if you have enough carbon monoxide or oxygen, the carbon monoxide binds on the surface, not just on the ends, and that makes a structure which is incompatible with existence in the fiber. So you get a hole. Eventually the holes get bigger, and the fiber fractures, which are the fractures that I demonstrated on previously. Okay, so that's so much for depolymerization.